Welcome back to Biology 2210 at CNM. We are talking about the nervous system. We're on chapter 12 in our textbooks. And we left off last time at objective number 111. And so what we know up to this point is what an action potential is. We're talking about neurophysiology. And so we just completed discussing the basic events of an action potential. Just to review, an action potential can only occur at places on the membrane where we have voltage-gated channels, and a typical neuron, that's called the trigger zone, which begins at the axon hillock at the base of an axon. In order to initiate an action potential, you have to get a variety of graded potentials known as excitatory postsynaptic potentials occurring at the dendrites or the cell body of the neuron. Those graded potentials will spread, weakening as they spread. If they're strong enough by the time they reach the trigger zone, and by strong enough we mean strong enough to depolarize the trigger zone from -70 to 15 millivolts less negative than that, to about -55, which is known as threshold, then voltage-gated sodium channels will open and we will initiate the action potential. When the voltage-gated sodium channels open in response to this voltage change, sodium floods into the cell membrane and that little tiny patch of membrane. This causes the inside of the cell membrane to become more positive. That is called depolarization. Those sodium channels start to close, which of course stops the depolarization, and then following the sodium-gated channels, the sodium voltage-gated channels, then potassium voltage-gated channels will open with their new threshold, higher threshold. That's why they open later. And potassium then will flood into the cell along its concentration gradient. And that, out of the cell along its concentration gradient. And so as potassium flows out of the cell, of course, that's a loss of positive charge. And so in that little patch of membrane where this is occurring, the cell membrane returns to a more negative polarity. This is called repolarization. And we actually overshoot resting in a phenomenon known as hyperpolarization. We make the membrane a little more polarized than -70. But in short order, that adjusts itself or fixes itself, and we eventually get back to resting where we began. So this is the events of an all-or-nothing phenomenon known as an action potential, which neurons use as signals to travel long distances through the body. In real time, of course, it's very rapid, appearing to be simply a spike, then with a relatively long return from hyperpolarization. So the spike component, the main elements of the action potential, occur in under 10 milliseconds, as you can see. And it takes maybe up to 40 or 50 milliseconds for the hyperpolarization to return to normal. So this is where we left off last time, and now we're ready to move on to objective number 112. And in objective 112, we talk about something known as the refractory period. A refractory period is a period of time during which the cell membrane is incapable of being stimulated again, or at least it's highly resistant to being stimulated by another stimulus. And this is known as the refractory period. And the refractory period occurs during the action potential. And there's actually two different types of refractory periods, known as the absolute refractory period and the relative refractory period. The absolute refractory period is the period of time during which, as I mentioned earlier, no stimulus will be able to trigger a new action potential on that piece of membrane. And it's not just that the membrane is resistant to being stimulated, it is incapable of being stimulated again during the absolute refractory period. There's nothing you can do to create a new action potential during the absolute refractory period. The reason this is because during the absolute refractory period, or the absolute refractory period is the period of time when the sodium gates are open, when these voltage-gated sodium channels are open and sodium is flowing into the cell. And again, the reason why there's a stimulus can't do anything further to stimulate the membrane is because the membrane is already experiencing an action potential. There's really nothing more you could do to stimulate that cell membrane, considering that the sodium voltage gated channels are already open. I mean, you can't do anything more than open the gates. So during this period of time when the sodium gates are open, there's really nothing additional you can do to stimulate the membrane, and so it's in what we call an absolute refractory period. The relative refractory period is a period of time during which the cell membrane is extremely resistant to stimulation. So it's not the same as the absolute. The absolute, there is nothing you can do to stimulate it. During the relative refractory period, you theoretically could stimulate the membrane again, but it would have to be an extremely strong stimulus to trigger a new action potential during the relative refractory period. So it is highly resistant to further stimulation. The relative refractory period begins after the absolute refractory period, and it occurs while the potassium gates are still open. And so at this point, the sodium gates have closed, but the potassium gates are still open, and they are allowing that potassium to flow out of the cell. But since the sodium gates have closed at this time, it is theoretically possible to open them again with an especially strong stimulus. So the membrane is resistant to stimulation, but it's not impossible. But it is highly resistant, and generally speaking, the membrane does not get further stimulated even during the relative refractory period. And so this relative refractory period lasts generally until hyperpolarization ends and the membrane returns to resting membrane potential. And again, remember, all of this, like the action potential, is highly localized. We're not talking about the entire cell membrane for the entire cell being in a refractory period. We're only talking about a tiny little piece of membrane where an action potential is occurring when we're talking about refractory periods. So again, like the action potential itself, refractory periods are very localized. Here we see a diagram, a graph showing the refractory periods. We see a typical action potential sketched out here with, again, millivolts on the y-axis and implied time axis on the x-axis. And so the absolute refractory period shown in reddish pink here, again, period of time during which there's nothing you can do to further stimulate an action potential. And again, this occurs while the sodium channels are open. Remember, we said most of the sodium channels close at this point where we start to get repolarization. But even in this repolarization phase, there are still a few sodium voltage gated channels that are still open. It's just that the potassium flow overwhelms the sodium, and we don't notice any effect of the sodium flowing at this point. And so the absolute refractory period lasts up to this point about roughly 10 milliseconds or so. And then the relative refractory period lasts until, again, we return to resting membrane potential. Remember, that occurs somewhere around 50 milliseconds total. Okay, so these are our two refractory periods. And as we'll see, there is some significance to these refractory periods. They actually play an important role in ensuring that the action potentials only move in one direction under normal circumstances in an axon. And we're going to come back to that in just a few minutes. So, how do action potentials move along an axon? Well, we've already hinted at this when we talked about action potentials in muscle cells. So the phenomenon is the same. Remember that we said in summary that action potentials are stationary, but they do constitute a signal that can travel along the axon. And we compared them to dominoes, each one tripping the other, creating a wave of energy that appears to be a traveling signal, even though each domino isn't moving. Well, It's a similar kind of situation in action potentials as we've discussed. This topic is referred to in your book as signal conduction in axons. I don't think conduction is exactly the best word to use here, because conduction does imply that something is flowing. The better term to use, and the term I'd like to use instead of conduction, is propagation. Signal propagation means that a signal is being propagated or created or repeatedly created along the length of the axon. And so I will use the term propagation instead of conduction because I do think it's more accurate. So remember, unmyelinated fibers do not have a coating or a myelin sheath that completely surrounds them. And so even though they sit in a groove in a Schwann cell or a oligodendrocyte, their cell membrane is exposed to the environment along their entire length. And so unmyelinated fibers have voltage-gated channels along their entire length. And so we're going to talk about propagation in unmyelinated fibers first, and then we will move on to propagation in a myelinated fiber. Excuse me. So remember, we don't get action potentials until the trigger zone, beginning with the axon hillock, because that's where the voltage-gated channels begin. And so when we get the first action potential at the trigger zone, of course, sodium enters the cell, and as we've repeatedly discussed, that sodium starts to diffuse, and it diffuses in all directions. It diffuses back toward the cell body, and it diffuses in the forward direction down the axon. Well, the sodium that diffuses in the forward direction toward the axon, of course, is going to encounter new or fresh voltage-gated channels. And so as it causes threshold levels of depolarization underneath those neighboring voltage-gated channels, we're going to get a new action potential created immediately adjacent to the original action potential. And so this is how the dominoes trip each other. Each action potential allowing sodium to diffuse into the cell, which will then diffuse a little bit downstream, exciting the next voltage gated channels and the next piece of membrane, and so on and so forth. And so this chain reaction continues until the nerve signal reaches the end of the axon. Again, just one action potential tripping the next one and the next one, and so on. This is referred to, by the way, as continuous propagation. Continuous propagation. Now, the question is, why doesn't this sodium that diffuses in and diffuses from the first action potential back to the cell body, why doesn't that cause action potentials to propagate in the opposite direction, back towards the cell body and the dendrites? Well, you may know the answer to this already. The simple answer is that, remember, the cell body and the dendrites do not have voltage-gated channels. And so even though the sodium from the first action potential is diffusing back towards the cell body and dendrites, we see no action potentials because of the absence of voltage-gated channels. So the action potential really only takes off, the falling dominoes really only take off in one direction from the trigger zone, and that is down the axon. So here we see a diagram, a visual, to help you visualize this. Again, the dendrites are going to pick up signals and creating excitatory postsynaptic potentials. Those will spread through the cell body. And again, if they're strong enough, if they're at threshold by the time they reach the trigger zone, we'll get the first action potential. And again, let's say the first action potential is occurring right here. Sodium is going to enter the cell. Remember that sodium is positively charged. That sodium will spread in all directions, both directions. Diffusion, remember, is not directional. Diffusion is random. So those sodium ions spread in all directions. The ones that spread in the forward direction are going to encounter, again, more voltage-gated channels in this immediately adjacent patch of membrane. and they are going to cause a threshold level of depolarization. We get a new action potential, and these new sodium ions that come in are going to diffuse, cause the next one, and so on and so on and so forth. But remember, the ones that spread in the backward direction are, from this trigger point, are spreading back into the cell body. Again, with the absence of voltage-gated channels, of course, we don't see any action potentials going in the reverse direction. Now, it may be interesting to ask, let's say I'm down in this part of the action, this part of the axon. Let's say the signal now has traveled this far, and I'm watching what happens just in this little region right here, which is what these pieces of the axon are actually supposed to be representing, is what's happening right here. I've got an action potential, say, occurring in a piece of axon. The sodium ions come in, and the sodium ions, again, they will spread. Just want to make it clear again that these sodium ions will spread in all directions, in the forward direction and in the backward direction. So I do have sodium ions diffusing backwards in this direction. But remember, they leak out of the cell relatively quickly, and the sodium-potassium pump is constantly at work pumping them out again. So they don't spread very far. They only spread a relatively short distance before they get pumped out or leak out. But of course, that's far enough to cause the next action potential, because that region of the membrane is right next to the original region. So we get the new action potential right there, right next to it. The question should be in your mind, since we're here out in the axon now, not back by the cell body, why don't these sodium ions that are spreading in the backward direction, why don't these cause an action potential? Thereby causing action potentials from a point out here to spread not only in the forward direction, but in the backward direction. Why doesn't this happen? Well, remember, the piece of membrane on this axon that is just in front of the current action potential is a piece of membrane that just went through its own action potential, right? In other words, this domino just fell in order to cause this one, which is falling right now. Well, if this piece of membrane just went through an action potential, notice it is still going to be in its refractory period. It won't be in its absolute refractory period, but it will still be in its relative refractory period, which, remember, lasts about 50 milliseconds. And so when this action potential is occurring, this piece of membrane just behind it is still clearly in the middle of its relative refractory period. And so the sodium ion that spreads back in the backward direction is not going to be able to stimulate this piece of membrane to experience another action potential because it just finished one and it is still recovering in essence. It is still in its refractory period. We don't get membrane that's not in its refractory period until we reach the green zone back here. But by the time the sodium spreads this far, remember, it has at this point probably leaked out. And again, just by diffusing across greater volume, it has diminished in its concentration. And remember, the sodium-potassium pump is also pumping it out using ATP. And so we've got all these reasons why the strength of depolarization is weakening, weakening, weakening the farther we spread. And essentially, by the time we get to the green zone, which could be stimulated, the sodium concentration just simply isn't enough to bring it to threshold. The only place where the sodium is high enough that it might cause an action potential is in this piece of membrane right next to the action potential. But again, since that is in its refractory period, there is no stimulation of an action potential. And so this is why once action potentials get out in the axon, there's really only unidirectional spread. It really only spreads in the forward direction towards pieces of membrane that have not been stimulated, at least not for a while. One last thing to point out here, technically an action potential is not the same thing as a nerve signal, though we often use the two synonymously. Remember, an action potential is a stationary momentary event. The nerve signal, what we call a nerve signal, is the wave of excitation, the wave of depolarization and repolarization that occurs as one action potential propagates the next and propagates the next and so on. So an action potential is stationary. A wave of action potentials is what we call a nerve signal. Now, propagation of the action potential is a little bit different in myelinated axons, because remember, myelinated axons have a myelin sheath around them, and so over much of their length they are actually covered by many layers of myelinated membranous Schwann cell or oligodendrocyte membrane, and that creates essentially an electrical insulation. Ions, sodium potassium ions, cannot flow quickly into and out of the axon if it's covered with all this myelinated membrane. So the only place where action potentials can occur is where the axon membrane is exposed to the environment, and that, recall, is in the gaps, the tiny little gaps between the adjacent Schwann cells or oligodendrocyte arms known as the nodes of Ranvier. The nodes of Ranvier, of course, contain many voltage-gated ion channels and are capable of experiencing action potentials. So what happens in a myelinated fiber is sodium enters the cell during an initial, say, action potential. Let's say it's happening at a specific node. And the sodium ions, once they get inside the cell, again, they start to diffuse away from the site of the action potential. Well, as they diffuse, of course, there's something to remember. They can't leak out of the cell because the cell, again, is covered in the neighboring in the adjacent regions to the node of Ranvier. It's covered with the myelin sheath. So not only does the myelin sheath prevent ions from coming in, it also prevents ions from leaking out. It also essentially incapacitates the sodium-potassium pump. The sodium-potassium pump cannot pump ions into and or out of the cell because of the myelin sheath. So these sodium ions that are injected during a single action potential, they start to spread and diffuse within the cell just as in an unmyelinated fiber, but their concentration doesn't diminish as rapidly as it would in a unmyelinated fiber because of the effect of the sheath preventing the loss of sodium. So as the sodium spreads away from the first action potential, it does start to fade a little bit just because it's spreading out through a greater volume. But that fading is very minor, it's very small. So the signal strength, the magnitude of the graded potential caused by these sodium ions does not start to fade very rapidly excuse me, as we move away from the node of Ranvier. And so as it turns out, by the time we get to the next node of Ranvier, there are still enough sodium ions that have been injected by the first action potential to create a threshold level of depolarization as far away as the next node of Ranvier. And at that point, when we get threshold depolarization at the next node of Ranvier, of course the voltage-gated channels open, and because the membrane is naked at that point, open to the environment, we can get ions flowing across the membrane, and we get a new all or nothing action potential at the next node. So instead of seeing many action potentials propagate each other like dominoes, as we would see in an unmyelinated fiber, it appears that when you look at myelinated fibers, you only see action potentials at nodes of Ranvier, and so the action potentials seem to leap or jump from one node directly to the next node, and then we don't see another action potential until the next node, and so on and so forth. So instead of seeing a continuous propagation of action potentials, we see them apparently jumping from node to node. All right, so once again, a visual of this, where again we have, say, an action potential occurring occurring right here. Sodium ions are injected. They flow in all directions. But because of the myelin sheath, the positive charge flows pretty rapidly. Remember, positive charges repel each other. So all these sodium ions are not just diffusing at normal rates, but they're actually repelling each other because positive repels positive, and they get pushed in this direction very rapidly underneath the myelin sheath. They do not leak out. They do not get pumped out. And so by the time they reach the next node, again, they're still pretty concentrated, or at least concentrated enough to cause a new action potential to occur now, where again the membrane is open to the environment and can experience ion flow. Again, what about the ions that spread in the reverse direction? Again, this spread, believe it or not, occurs so rapidly due to the repulsion of the positive charges against each other that they arrive at the previous node very quickly, just like they arrive at the next node very quickly. And remember, this node, this prior node, just went through its own action potential to create this action potential. And so guess what? This membrane in this prior node, again, is still in its relative refractory period. And so it is not, this signal is not going to be strong enough to initiate an action potential in the backward direction, again, for the same reasons. What about 2 nodes or 3 nodes prior? Well, those nodes way over here might be out of their refractory period, but by then the distance that has been spread is so far that in fact the sodium ions have diminished in concentration enough to not be adequate for threshold level depolarization. Because remember, as they spread back to this first node, even though this membrane is in its refractory period, the sodium that does diffuse back this far will start to leak out and will start to get pumped out by the sodium-potassium pump. So by the time you get past this first node, there's really not enough sodium left to go on to stimulate any prior nodes that might be out of their refractory period by this time. So once again, the only direction we have to go is forward. And again, this diffusion is much more rapid than would be typical diffusion because of the repulsion of the positively charged ions against one another. And so the action potential would occur here, and then we would have another action potential occurring at the next node. So one occurs here, and then the next time we would see an action potential is right here. So if we weren't really paying attention to the flow of sodium within the axon, if we were just looking at it from outside the axon, it would appear that the action potential jumped from one node and then magically appeared at the next node, and then of course will magically appear at the next node. And so you get this kind of jumping effect where the action potential appears to leap or jump from node to node. This is referred to as not continuous propagation, but saltatory propagation of a nerve signal. Saltatory propagation. And a saltation is a fancy word for a leaping or a jumping. I just want to point out that saltatory propagation is faster than regular continuous propagation, because in continuous propagation, each action potential must trigger an action potential in the neighboring membrane, which then must trigger an action potential in the neighboring membrane, and so on, and so on, and so forth. Again, the dominoes have to trip each other directly, one after the other, one after the other, one after the other. And so you note that to travel this distance between one node and the next, or whatever that distance would be in an unmyelinated fiber, it would take many action potentials to get this far. And even though they are very rapid, they still, each one still takes some time to perform, right? At least 5 milliseconds to 10 milliseconds for the spike part alone. And so that adds up. Over time, the time it takes for each action potential to occur does add up. Whereas here in saltatory conduction, you only have two action potentials traveling the same distance. And so that's one reason why the speed of propagation is faster, just have fewer action potentials occurring. And then secondly, remember, this flow of sodium is much faster in the myelinated sheath because of this high concentration of sodium that can't leak out. And so these sodium ions repel each other much more than in an unmyelinated fiber, and so they move much more quickly than typical diffusion of sodium ions in an unmyelinated fiber. And so for those two reasons, we see that saltatory propagation is going to be significantly faster than continuous propagation. And you may remember one of the things we said about the myelin sheath, one of its functions was that it affected the speed of nerve signal propagation. And we said that myelinated fibers can conduct or propagate nerve signals more rapidly than unmyelinated fibers. And now we understand why. All right, here's the book's version of what I was trying to draw with saltatory propagation. The action potential is in pink, and it again appears to leap from one node to the next, and of course it would continue on to the next and the next and the next. There is a continuous flow of something, but it's not an action potential, it's a continuous flow of sodium underneath the myelin sheath. Okay, so that completes our discussion of action potentials. Now we're ready to move on to the topic of synapses. And again, this is a little bit of review because we looked at a synapse in some detail when we covered the muscle system and we looked at the neuromuscular junction. The neuromuscular junction is a type of synapse, and it's a very typical type of synapse. But what we'll learn in this section is that not all synapses behave exactly the same way as the neuromuscular junction does. But we, having looked at the neuromuscular junction already, does equip us with some basic terminology that we will review here. So remember, action potentials or nerve signals are great for carrying messages long distances along an axon. However, action potentials and electrical nerve signals are not good for carrying the message from one cell to another. And it doesn't matter whether that second cell or that next cell is another neuron or a muscle cell or a gland. We have to have another way for the signal to travel from the end of the axon to the next cell. And essentially, that's what a synapse is. The synapse is the functional connection between a axon, the tip of an axon, and the next cell that it's trying to stimulate. Again, be it a neuron, a muscle cell, or a gland. As we know from the neuromuscular junction, it's a chemical signal that traverses the gap or the space between the axon terminal and the next cell, and this chemical is referred to as a neurotransmitter. And the gap over which the neurotransmitter has to diffuse in order to stimulate the next cell is called a synaptic cleft. And as we know, once that neurotransmitter binds somehow to the next cell across the synapse, that's going to create new action potentials, hopefully or potentially, in the next cell, or it's going to stimulate the next cell somehow. In the case of a glandular cell, of course, there is no subsequent electrical activity. In that case, we're stimulating the gland to secrete something. So it doesn't always stimulate new electrical activity in the next cell. It depends on what the next cell is. When we're talking about a synapse specifically between two neurons, of course, the next cell will potentially create new electrical activity. And we use terminology that I introduced earlier in this chapter, but I'm just going to review it again. If I have a synapse between two neurons, then the first neuron in the pathway, the one that's going to deliver neurotransmitter from its axon, is referred to as the presynaptic neuron. In other words, that's the one in front of the synapse. So again, this is the one releasing neurotransmitter onto the second neuron. That second neuron we refer to as the post-synaptic neuron. In other words, it is after the synapse, and it's going to pick up or bind to that neurotransmitter. And again, if the postsynaptic potentials are strong enough, that may trigger a new action potential in this postsynaptic neuron, which will then carry its message on to its target cell, which again might be another neuron, muscle cell, or gland. Now, as we said, in the case of a neuron-neuron synapse, which is what we're going to focus on from this point out, just neuron-neuron synapses for the most part. The presynaptic neuron, when it synapses with the postsynaptic neuron, can synapse really with any part of that postsynaptic neuron. It's very common. Probably the most common scenario is for the presynaptic neuron to synapse with a dendrite of the postsynaptic neuron. This is referred to as an axodendritic synapse. Those are the ones we usually think about. But there are other types. It is possible for the presynaptic neuron to synapse with the cell body of the second neuron or postsynaptic neuron directly and not synapse with a dendrite. That kind of synapse is called an axosomatic synapse, where the axon terminal is synapsing with the cell body of the postsynaptic neuron. And there are even synapses known as axoaxonic synapses. In an axoaxonal synapse or axoaxonic synapse, the presynaptic axon is synapsing with the axon of the postsynaptic neuron. So an axon synapsing with an axon of another neuron. This may seem unusual to you, but as we'll see later in this chapter, it's one way that a neuron, a presynaptic neuron, can influence the behavior of a postsynaptic neuron, either increasing the likelihood of it firing, or in some cases inhibiting or decreasing the likelihood of that postsynaptic axon from firing. But we'll come back to that in a little bit. All right, so an individual presynaptic neuron can have a huge number of synapses. A spinal motor neuron, remember, for instance, can be involved in a tremendous number of synapses if it's a large motor unit. Right? Remember, a motor unit was a single axon in all of the skeletal muscle cells that it innervates. And so, a single axon can branch many times, allowing a single neuron to have many, many, many neurons at its axon terminals. Also remember that a single multipolar neuron is going to have many dendrites. Potentially, as you see here, as many as 8,000 dendritic tips that can become involved in a synapse. Each one can be involved in a synapse. And so you've got lots of potential branching at both ends, both the dendrites especially, and in the axon. And don't forget that the neurosoma, the cell body of a neuron, can also be receptive and be involved in synapses. And so in some cases, in the most extreme cases, you have some neurons, say in the cerebellum, for instance, that can have as many as 100,000 synapses per cell, per individual cell. Can be involved in as many as 100,000 synapses. So this synaptic network in the brain is truly astounding in terms of the number of connections that can exist inside the brain because of all these synapses that each individual neuron can become involved in. And so here we see a diagram again of the various types of synapses: axodendritic, axosomatic, right, axon onto the body, and then axoaxonal, which is usually downstream quite a bit, where an axon synapses with usually near the distal end of another axon. And again, we'll come back to that to talk about how this affects the postsynaptic behavior. Well, as we said, the synaptic cleft is the gap across which the neurotransmitter has to diffuse in order to stimulate the postsynaptic cell. In the nervous system, most synapses are chemical synapses, again relying on neurotransmitters. Remember, there are such things as electrical synapses, and what we mean by electrical synapses are gap junctions. Remember, gap junctions are special junctions that allow materials to diffuse directly from one cell to another. So cells that are hooked up by gap junctions are essentially electrically connected because the sodium that gets injected in one action potential in one cell can spread through the gap junction directly into the next cell, causing threshold depolarization in the next cell and a new action potential. These are referred to as electrical synapses, and again, you do see them in some places. Some neurons in the body are connected by gap junctions. We talked about astrocytes having some gap junctions between them. We talked about cardiac cells being electrically connected all to each other through gap junctions, and we also mentioned previously single unit smooth muscle cells being connected via gap junctions. The advantage of gap junctions and electrical synapses, of course, is very quick transmission. We don't experience much of what we call synaptic delay. We'll talk about synaptic delay towards the end of this chapter, but it does take time to stimulate the release of neurotransmitter, have the neurotransmitter diffuse across the synaptic cleft, bind, open channels, and so on. That all, again, is very quick, but it does take a certain amount of time. We don't have to do any of that in an electrical synapse, so they tend to be quicker in their cell-to-cell transmission. The disadvantage is that they are difficult to regulate. With chemical synapses, we can regulate intensity. We can cause a variety of different kinds of effects, but electrical synapses are generally all or nothing phenomena, and they're more difficult to regulate. So they're quick, but we can't get involved in the kind of integration and regulation that we need usually in an organ like the brain. Now, in a more typical chemical synapse between neurons, of course, we see the familiar scenario of the presynaptic neuron containing synaptic vesicles, which contain neurotransmitter. Just to point out, in the neuromuscular junction, we always talked about the neurotransmitters being acetylcholine. As we will learn later in this chapter, acetylcholine is just one of many possibilities for neurotransmitter molecules. So not all synaptic vesicles contain acetylcholine. They do contain neurotransmitter, but there are a variety of types, as we will learn in the next video. Many vesicles, as you can see, are docked on release sites on the plasma membrane at the axon terminal. So when you get to the axon terminal, what you see is that many vesicles are sitting just on the inside of the axon terminal's cell membrane. In other words, they're ready to go. They're like locked and loaded, ready to go. All you have to do is alter membrane fluidity just a bit, and those vesicles that are essentially sitting attached to the inside of the axon terminal cell membrane will fuse with the membrane and release their neurotransmitter. We do have reserve synaptic vesicles that are located farther away from the surface of the axon terminal, and as the name implies, we hold those in reserve to replace neurotransmitter that we lose as synaptic transmission takes place. Remember, the postsynaptic membrane, like the motor end plate of the muscle cell, but in this case it's again the postsynaptic neuron membrane, contains membrane embedded proteins that function as receptor molecules. And remember, these receptors are often chemically gated channels. Your book calls these ligand regulated ion gates. I call them chemically regulated ion gates that usually either open or close in response to binding by the neurotransmitter. Here we see a micrograph of a chemical synapse, and here we're seeing the axon terminals of the presynaptic neurons. These are the axon terminals, the little terminal boutons or buds. Excuse me, and this is the cell body of a postsynaptic neuron. So what you're seeing here would be an axosomatic synapse, not onto a dendrite, but directly onto the body of the postsynaptic neuron. It's interesting to see that what these things actually look like. Very similar to what we've been describing. You can also see why in the old days we used to think that there was a physical connection between pre and postsynaptic cell. We used to think that they physically touched, because it certainly looks like they're actually in physical contact. But remember, there is, even though they look like they're touching, there is a tiny little gap, a synaptic cleft between them. Here we see a familiar diagram of what we've been talking about. Again, the axon terminal with the synaptic cleft between it and the postsynaptic neuron. All the presynaptic, all the vesicles in the presynaptic axon terminal docked in position, waiting to be released upon proper stimulation, which is an action potential that comes down the axon. Neurotransmitter is released, and we do not see synaptic vesicles in the postsynaptic neuron. Of course, there's no need for them there. Instead, we see a membrane full of receptor molecules embedded in the membrane, just as we described. Excuse me. Here we see a conventional histological view of a synapse. This one is specifically the neuromuscular junction between the axon terminal and the muscle fiber. So here's the axon terminal, and then of course this big pink cylinder would be the muscle cell. Another neuromuscular junction picture. This is electron micrograph, again showing how very, very tiny the synaptic cleft is between these two cells. Yet another picture. I'm not going to belabor it at this point. Okay, so let's talk a little bit about neurotransmitters at this point. Again, neurotransmitters are, as we know, molecules that are released from synaptic vesicles when a nerve signal arrives at the axon terminal, the knob, the so-called synaptic knob, which is that swollen tip of the axon. These neurotransmitters, again, bind to receptors on another cell, and we usually think of them, again, as creating electrical activity in the postsynaptic cell, but sometimes, again, it just alters the cell's physiology. That's most obvious, again, in the case of glands, where we don't stimulate a new action potential. We stimulate the gland physiologically to do something like manufacture or release a secretion. As I've mentioned, acetylcholine is not the only neurotransmitter, though it's the only one we've really talked about much this semester. But you'll notice here, more than 100 different chemicals have been identified as having neurotransmitter function. So a wide variety of chemicals have been identified as neurotransmitters in the body. Fortunately, most of those neurotransmitters fall into four major chemical categories. And the four major categories of neurotransmitters are, as you can see here, and in Objective 131, acetylcholine, amino acids, monoamines, and neuropeptides. So, as you can see, many of the neurotransmitters are not necessarily very large molecules. Amino acids, remember, are building blocks of proteins, and while they're not ionic in size, or not as small as water, they're certainly not macromolecules by any means. So let's talk about each of these classes in order. We're going to start with acetylcholine, our familiar friend from the neuromuscular junction. Acetylcholine, again, as we've said here, it is in a class by itself. Acetylcholine is formed from acetic acid and a substance known as choline, and pretty much that's it. Fairly simple structure. Amino acid neurotransmitters, as the name implies, are essentially amino acids or slightly modified amino acids. Now, remember, there are 20 different amino acids that we use in the body to make proteins. Not all 20 of these amino acids function as neurotransmitters, but a few do, and some of the more common ones are glycine, glutamate, aspartate or aspartamine, and gamma-aminobutyric acid, which of course is a mouthful, and most people simply just abbreviate gamma-aminobutyric acid as GABA, gamma-aminobutyric acid. The monoamines are even smaller than the amino acid neurotransmitters. Essentially, monoamines are made from amino acids, and they are made by removing the carboxyl group. Remember what a carboxyl group is from introductory biology. It's I'll see if I can draw one here, a carbon double bound to an oxygen and bound to a hydroxyl group. And so every amino acid has an amino group at one end and a carboxyl group at the other end. And so if we pull off the carboxyl group, what we have left is something called a monoamine. A monoamine, so slightly smaller than a typical amino acid. These are fairly familiar. Monoamines are some familiar molecules, and some even familiar as neurotransmitters. So the catecholamines is one group of monoamines, including perhaps the best known neurotransmitters after acetylcholine. Those include epinephrine and norepinephrine, also known, by the way, as adrenaline and noradrenaline. So these are neurotransmitters and hormones. Epinephrine also functions as a hormone that are associated with the fight or flight response. Dopamine is another catecholine, very similar. Dopamine is a brain neurotransmitter involved in control of muscle function. Other types of monoamines in the body that also function as neurotransmitters, surprisingly. Histamine, which we often associate with inflammation. It's an inflammatory chemical, but it can serve a neurotransmitter function in the central nervous system. ATP, which we think of as a energy fuel source, but can function as a neurotransmitter. And serotonin, which is also a neurotransmitter in the brain that seems to be associated with mood and some basic emotions. Purines are another type of neurotransmitter. That include ATP and adenosine, which is the precursor to ATP. It's basically ATP without any of the phosphates. These are modified types of monoamines. And remember, purines are building blocks, one of the building blocks of DNA and RNA. They're one of the nitrogenous bases. And then we also have neuropeptides, as we mentioned, as the fourth major category of neurotransmitters. Neuropeptides are among the largest of the neurotransmitters because, remember, a peptide is 2 or more amino acids linked together. And so, as you see, we have chains of 2 to 40 amino acids. Now, even with 40 amino acids, that's not a huge protein by protein standards, but it is going to be quite a bit larger than the other neurotransmitters. Some neuropeptide type neurotransmitters include things like cholecystokinin. In A&P2, you'll talk more about cholecystokinin as a hormone of the digestive system released by, for instance, the small intestine to help regulate digestive activity, but it can also function as a neurotransmitter in some cases. And substance P is another neuropeptide. Substance P is a neurotransmitter that we find in the spinal cord, and it is seemingly involved in the modulation of pain signals coming in from the body. And so substance P is involved in pain suppression, and we use some drugs to influence substance P and therefore help reduce pain. And again, some of these function as hormones as well. And I mentioned cholecystokinin, of course, which is associated with the digestive system. Now, those are the four main chemical categories to which neurotransmitters belong. We did throw in an extra one, the purines, in there, but there aren't too many purines. Just to be complete, we also want to mention that there are some gases. Well, there are some gases that can function as neurotransmitters as well, in a way. Nitric oxide is one, and believe it or not, carbon monoxide is also one. Now, notice these are inorganic. Remember, to be organic, a molecule has to contain both carbon and hydrogen, and you see neither one of these contains hydrogen. This nitric oxide doesn't contain carbon or hydrogen. So while the vast majority of neurotransmitters are organic, in this case we've got a couple of inorganic neurotransmitters. These are not stored in synaptic vesicles, but they are synthesized by presynaptic neurons, and they tend to diffuse out of the axon terminal relatively freely rather than being released under some kind of tight control by action potentials. But they do function like neurotransmitters, because as they diffuse across the synaptic cleft, they can bind to receptors on the postsynaptic cell and cause effects. And so they're, I don't know if I want to call them neurotransmitters or neurotransmitter-like substances. They are, interesting to note, a couple of gases that are made and released by the presynaptic cell that can have an influence on the postsynaptic cell. And here again we see our categories, some structural formulas or diagrams for these different categories. Again, acetylcholine all by itself, made from acetic acid and choline, linked right there. Amino acids, remember, a typical amino acid has a carboxyl group at one end and an amino group at the other, and then a variety of possible side chains or structures in between the carboxyl and the amino group. And so all of these have that basic structure. Monoamines are a variety of structures where, again, we've pulled off the carboxyl group and essentially replaced it with something else. So here's your typical amino group, and then the central part of an amino acid, and then instead of a carboxyl group like you'd see in a typical amino acid, we've replaced it with something else. It usually involves a six-sided ring. These are referred to as the catecholamines. And then some related monoamines like serotonin and histamine. Again, an amino group, a central portion, which is reminiscent of an amino acid, but then instead of a carboxyl group, again, we just have something else in that position. And then, as we said, the neuropeptides, which are the largest of the neurotransmitters, with their strings of amino acids. Each one of these little spheres would be something akin to this structure, right? So we're putting a bunch of these together to make a short peptide chain. These are the neuropeptides that we mentioned, like substance P in the spinal cord. Enkephalins. Enkephalins are pleasure neurotransmitters. They seem to be associated with certain feelings of satisfaction For instance, runner's high and the sensation of satisfaction you get with working out and physical activity. Those are endorphins and enkephalins, which are neuropeptides, endorphins and enkephalins. All right, now as we said, synapses are not all created equal, so to speak. There are different types of synapses in the body and different ways in which neurotransmitters can influence the postsynaptic cell. So different neurotransmitters and different effects of those neurotransmitters create different kinds of synapses. So what do we mean by different effects of neurotransmitters? Well, some neurotransmitters are excitatory, meaning they cause a depolarization in the postsynaptic membrane. Others can be inhibitory, and remember, by inhibitory we mean that the neurotransmitter creates not a depolarization but a hyperpolarization in the postsynaptic membrane. And of course, different neurotransmitters can have different effects. One can be excitatory, one can be inhibitory. But what's also interesting to note is that a single neurotransmitter may have different effects based on the type of receptor in the postsynaptic cell. So epinephrine, for instance, norepinephrine, which is the more common of the two, norepinephrine as a neurotransmitter might excite one cell but inhibit another. It's the same neurotransmitter, so how can it have opposite effects and cause depolarization in one case and hyperpolarization in the other? Well, it really all depends on what kind of receptor for norepinephrine the postsynaptic cell has. So one neurotransmitter can have both kinds of effects depending on what's going on with the postsynaptic cell membrane. Also, we've been talking as if all neurotransmitters, as they diffuse across the synapse, we've been talking as if they always bind to a chemically gated ion channel when they arrive at the postsynaptic membrane. Well, that's not 100% true either. Many of them do. Acetylcholine did, for instance, at the neuromuscular junction, caused a motor end plate potential, a kind of graded potential by opening ion channels. But they don't all do that. And so there are some neurotransmitters, as we'll see, that will diffuse across the synaptic cleft and bind to a receptor molecule that isn't an ion channel. The receptor molecule, as we'll see, may then stimulate a second molecule, which may stimulate a third molecule to be released within the postsynaptic cell and cause some physiological effect or membranous effect in the postsynaptic cell. So sometimes there are what we call second messenger systems through which the neurotransmitter acts. So the neurotransmitter itself doesn't cause the immediate response, but it triggers a series of activations which lead to second messengers, which then will have the direct effect on the postsynaptic cell. So we're going to see examples of this, of all these different effects, by looking at three basic kinds of synapses that exist. Excitatory cholinergic synapses, inhibitory GABAergic synapses, and excitatory adrenergic synapses. And there are other kinds, of course, because again there's over 100 different types of neurotransmitters, but we're going to use these three to illustrate the various kinds of events that can happen at different kinds of synapses. All right, we're just about at 1 hour and 3 minutes at this point, so we're going to stop here. In terms of the objectives, we have made it up to objective 136 for today. Next time we'll pick up with objective 137, and I don't doubt that we'll be able to finish chapter 12 next time. All right, so until then, stay safe.